Welcome back to the deep dive. Today, we are making a pretty significant geographical shift in our microbial journey.
We really are.
We are leaving the warm, anaerobic, nutrient-rich comfort of the human gut, the world of E. coli, and the Enterobacteriaceae we've come to know and love.
Right. And we are stepping out into the cold, harsh, and strangely moist environment of the hospital ward.
It's a completely different battlefield.
It is a distinct shift in mindset. You really have to reset your expectations. We are leaving the fermenters behind and entering the world of the, quote, non-fermenters.
Of non-fermenters.
We are talking about organisms that don't just live in the patient. They live in the sink next to the patient. They live in the mop bucket, the ventilator tubing, and, the vase of cut flowers at the nursing station.
The survivors, the opportunists.
Exactly. We are diving into Murray's Medical Microbiology, Chapter 27. The focus is on Pseudomonas, aeruginosa, and what I like to call the opportunistic alliance.
The opportunistic alliance. I like that. Who's in this alliance?
Well, you've got related bacteria like Burkholderia, Stenotrophomonas, Acinetobacter, and Moraxella.
It sounds like a faction in a dystopian novel. But looking at the source material, the theme here really is opportunistic resilience.
That's the perfect term for it.
These aren't the primary predators that hunt down healthy people, right? These are scavengers.
That's the conceptual anchor. Unlike, say, Salmonella or Shigella, which are professional pathogens designed to breach healthy defenses, these non-fermenters are largely environmental organisms. They're saprophytes.
So they just live out in the world.
Right. They live in soil, decaying vegetation, water. Honestly, they don't want to kill you. They just want to survive. But if you give them an opening, a chink in the armor, like a burn, a catheter, cystic fibrosis, or intubation, they will invade with ruthless efficiency.
Before we get into the specific crimes of these bacteria, let's establish the biological lineup. We keep saying non-fermenters. Why does that metabolic detail matter so much to a medical student or a clinician?
It matters because it tells you where to look for them and how to identify them in the lab. The Enterobacteriaceae, the gut bugs, are facultative anaerobes that ferment glucose.
Right. They can live with or without oxygen, but they're happy to ferment.
They love the low oxygen environment of the colon. The organisms in Chapter 27, primarily, are oxidizers. They're obligate aerobes. They rely on aerobic respiration.
So they need oxygen. No exceptions.
For the most part, yes.
Yeah.
They utilize something called cytochrome sac oxidase in their respiratory chain. And this is why you won't typically find them causing, say, abscesses deep in the belly where there's no oxygen.
So where do you find them?
You find them in the lungs, in wounds exposed to air, and on surfaces, anywhere they can breathe.
And this metabolic difference leads us to the first big branching point in the diagnostic algorithm, doesn't it? The oxidase test.
Correct. This is extremely high yield. If you have a gram-negative rod, the first question is it an Enterobacteriaceae or a non-fermenter? You do the oxidase test.
And what does that tell you?
If it's E. coli or Klebsiella, it's oxidase negative. The region stays colorless. But if it turns a deep purple, meaning it's oxidase positive, you are very likely looking at Pseudomonas or one of its cousins.
Purple means specific trouble. Okay, let's introduce the leader of this alliance. The organism that Murray devotes the most pages to. Pseudomonas aeruginosa.
The head honcho, the kingpin. Pseudomonas aeruginosa is the quintessential opportunist. It is absolutely ubiquitous in the environment.
If we were building a criminal profile for this Bugga Most Wanted poster, what does it look like under the microscope?
Well, visually, it's a small gram-negative rod. But Murray mentions a specific morphological detail that really aids in pattern recognition. They're typically arranged in pairs.
In pairs. I've heard the memory hook that they look like 2 red grains of rice end to end.
That's a very helpful visualization. It's quite distinctive. So you see these little red pairs on the gram stain, and you get a positive oxidase test.
The picture is starting to come together.
But Pseudomonas is unique because it engages more than just your sense of sight. It engages your sense of smell.
The olfactory diagnosis, I've heard about this.
Indeed. It has a very, very distinct olfactory signature. It produces a sweet, almost grape-like odor.
A grape-like odor from a bacterium?
Yes. Specifically, it produces a compound called aminoacetophenone. It's very noticeable.
I've seen some other sources, including Javet's, describe it as smelling like corn tacos or tortillas.
It's a spectrum of sweet and carb-heavy. I think that's a good way to put it. Whether you smell grapes or maybe fruity candy or corn tortillas, the takeaway is that it is distinct.
So if you're on the wards.
If you walk into a burn unit or an ICU room, and you hit a wall of that sweet, fruity air, your alarm bell should be ringing for Pseudomonas.
So we have the look, we have the smell. Now let's talk about the color, because Pseudomonas is arguably the most colorful pathogen we discussed.
It is an artist. It really is. Pseudomonas aeruginosa produces these water-soluble pigments that diffuse into the growth medium, or, more clinically, into the patient's bandages.
And there are two main pigments we need to know, right?
Yes, two main pigments you need to understand, both for exams and for pathophysiology. They have very specific roles.
We've got pyocyanin and pyoverdine. Let's break them down, because the names matter here.
They absolutely do. Let's start with pyocyanin. The root cyan refers to blue, like cyan ink. This is a blue pigment.
And is that unique to Pseudomonas?
Crucially, yes. Pyocyanin is unique to P. aeruginosa. No other species produces it. If you see that blue pigment, you've made your diagnosis.
But is it just for show? Or does it do something?
Not at all. It's a virulence factor, a potent one. Pyocyanin catalyzes the production of toxic oxygen forms. We call them reactive oxygen species, or ROS.
Like superoxide and hydrogen peroxide.
Exactly. It essentially generates a cloud of toxicity around the bacteria. It's a form of chemical warfare.
How does that help the bacteria?
Well, it does two things. First, it stimulates an inflammatory response, which attracts our neutrophils to the site. But then the ROS it's generating actually damage and kill those very neutrophils and the surrounding host tissues.
So it calls in the cavalry and then shoots them when they arrive.
It creates a diversion and destroys the attackers simultaneously. It's diabolically clever.
That's the blue pigment, pyocyanin. Then we have pyoverdine.
Pyoverdine is yellow-green. The name comes from verd, like verde, meaning green. This pigment has a totally different job. It's a cytophore.
A cytophore. Let's unpack that term. I think it's important.
It is. So you have to think of the host environment as a battlefield where iron is the most precious resource. It's like oil.
And our body tries to keep it locked up.
Cries very hard. The human body sequesters iron in proteins like ferritin and transferrin specifically to starve invading bacteria.
So the cytophore is the bacteria's answer to that.
It's the bacteria's secret weapon. Pseudomonas releases pyoverdine, which goes out and binds iron with incredibly high affinity, stronger than our own proteins can hold it, and it hauls it back to the bacteria for use.
It's an iron thief.
A very efficient one. And together, they create a distinct color palette. If you mix the blue pyocyanin with the yellow-green pyoverdine,
You get green, blue plus yellow makes green.
Precisely. This is the reason why Pseudomonas infections are classically described as producing green pus, or why the colonies on an agar plate often have a beautiful metallic green sheen.
So as a clinician, if you see green drainage from a surgical wound or a burn,
You don't need to wait for the lab report to start thinking Pseudomonas. It's a massive clinical clue.
that is a critical clinical anchor. Green pus equals Pseudomonas. Now, we've established that this bug is everywhere and it's colorful, but why is it so hard to kill? Murray describes it as having simple nutritional requirements. That sounds like a massive understatement.
It's the MacGyver of the bacterial world. It is not a picky eater. It can utilize over 80 different organic compounds for carbon and nitrogen,
80.
at least. It can grow in distilled water if there are just trace minerals leached from the glass container. It can grow in some disinfectant solutions, particularly quaternary ammonium compounds, if they aren't formulated perfectly.
That is terrifying. It can live in the thing designed to kill it.
It's why it persists in hospital sinks, mop heads, and respiratory therapy equipment. It thrives in moisture and needs almost nothing to survive.
So we have the stage set. We have a resilient, green, grape-smelling, moisture-loving fortress of a bacterium. Now let's talk about its weaponry. Murray breaks down the virulence factors into a massive arsenal. It's not just one toxin. It's a combined arms approach.
I like to view Pseudomonas as a medieval fortress that is also a tank. It has defensive walls and it has offensive artillery.
Let's start with how it gets a foothold, the grappling hooks, the adhesins.
Right. Sticking to the target. If it can't stick, it gets washed away by the lungs and mucociliary elevator or the flow of urine. So Pseudomonas uses flagella for motility. It can swim to the site.
Okay, so it's mobile.
And then it uses pili for adhesion, specifically type 4V pili, which allow for something called twitching motility, where it literally crawls along surfaces to find the best spot.
And once it sticks, it bunkers down. This brings us to the alginate capsule. This is probably the single most important concept for understanding chronic Pseudomonas infection, especially in one particular disease.
It is the centerpiece of the cystic fibrosis, or CF, story. Alginate is a mucoid exopolysaccharide. Basically, it's a thick, sticky slime layer.
And this is where the phenotype switch happens, right? It's not always slimy.
Exactly. When Pseudomonas first infects a lung, it's usually non-mucoid. It doesn't have that thick capsule. But in the unique inflammatory environment of the CF lung, it undergoes a genetic switch.
What triggers that switch?
It's the chronic inflammation, the thick mucus. The bacteria senses this environment and says, I need to build a fortress to survive here. So it starts overproducing this alginate, becomes mucoid.
Mucoid is such a visceral word. What does that look like in the lab?
It looks like snot on a petri dish. The colonies are huge, glistening, and run together. But in the lung, that alginate matrix forms a biofilm.
And a biofilm is more than just slime. It's a community.
It's a city. Think of a biofilm as a concrete bunker. The antibodies can't penetrate it. The phagocytes, our white blood cells, they literally cannot engulf the bacteria because they are embedded in this thick slime.
And what about our best weapons, the antibiotics?
They diffuse into the biofilm very slowly, often failing to reach lethal concentrations at the bottom layers. So the bacteria are sitting inside this bunker, protected from the immune system and our drugs, just replicating.
And they aren't just sitting there passively, they are talking to each other.
Quorum sensing. This is a fascinating aspect of Pseudomonas biology. It's a system of cell-to-cell communication.
How does that work?
When the population density gets high enough within that biofilm, they start releasing small signaling molecules. When the concentration of those molecules hits a certain threshold, it tells all the bacteria in the community.
We have enough numbers.
Okay, guys, we have enough numbers. It's time. Turn on the toxin genes. Ramp up the virulence. It's coordinated warfare triggered by population density.
That's the defense. Let's look at the offense, the heavy artillery. The biggest gun in the arsenal is exotoxin A, ETA.
Yes. If you are a student preparing for a board exam, underline exotoxin A in your mental notes. This is a classic.
So how does it work? What's the mechanism?
It disrupts protein synthesis. It's an absolute showstopper for any eukaryotic cell it enters.
Okay.
It enters the host cell, and it catalyzes the ADP ribosylosis of elongation factor 2, EF2.
Okay, let's unpack that mechanism. So the ribosome is the protein factory. EF2 is the worker on the assembly line that's physically moving the peptide chain along.
Exactly. It's essential for that translocation step. And exotoxin A essentially freezes that worker. It covalently attaches an ADP ribose group to it, which inactivates it completely. It shuts down the assembly line.
And if a cell cannot make proteins, it dies.
It dies. It's that simple. This is what leads to the profound tissue necrosis that we see in these infections.
Now, this mechanism sounds incredibly familiar. Is there another bug we've covered or will cover that uses this exact same mechanism?
Diphtheria toxin, from Corium bacterium diphtheria. It produces a toxin with the exact same molecular mechanism. They both target the same protein, EF2, in the same way with ADP ribosylation.
So for an exam question, you'd have two different bacteria, two structurally different toxins, but the same murder weapon.
Correct. The clinical context would be the differentiator. And in Pseudomonas, this toxin contributes to the dermatonecrosis, the skin death and burn wounds, and the corneal damage in eye infections.
But exotoxin A isn't the only weapon. Pseudomonas also produces enzymes that act like biological bulldozers.
It is a destroyer of tissue scaffolding. It produces a family of enzymes called elastases, specifically LASA and LASB.
And as the name implies, elastase breaks down elastin.
And where is elastin a critical structural protein? In your lung parenchyma to keep it stretchy and allow you to breathe.
And in blood vessels.
And crucially, in your blood vessel walls. They are rich in elastin.
So if you break down the blood vessel walls,
you get hemorrhage and you get invasion. Pseudomonas doesn't just stay on the surface. It uses elastase to chew through the vessel wall and enter the bloodstream, causing sepsis. This is why Pseudomonas skin lesions, which we'll discuss later, are often hemorrhagic or black. The blood supply has been destroyed.
And there's one more weapon system we need to cover, the molecular syringe, the type 3 secretion system.
This is a marvel of evolutionary engineering. It's a needle-like structure on the surface of the bacterium. When the Pseudomonas comes into direct contact with a host cell, like a macrophage or an epithelial cell,
it docks with it.
It docks.
Yeah.
And it inserts this needle directly through the host cell membrane.
It injects poison straight into the room.
It bypasses the extracellular space entirely. It doesn't need a receptor. It injects effector proteins, exoenzymes S and T, directly into the cell cytoplasm.
And what do those do inside the cell?
They are toxins that disrupt the cell's internal structure. They rearrange the actin cytoskeleton, which causes the cell to collapse, or they can trigger apoptosis, which is programmed cell death. It's an immediate, targeted assassination of an immune cell or an epithelial cell.
So let's recap the arsenal before we move to resistance. It swims with flagella, sticks with pili, builds a slime bunker with alginate, stops your protein synthesis with exotoxin A, dissolves your connective tissue with elastase, and injects poison directly into your cells with a type 3 syringe.
It is a formidable enemy, a multifaceted threat.
And we haven't even touched on the trait that makes it a daily headache for pharmacists and infectious disease doctors: antibiotic resistance.
The unbeatable shield. This is why Pseudomonas is such a clinical nightmare.
So is it just that it learns quickly, or is it born tough?
It's both. It has significant intrinsic resistance. Its outer membrane is like a very strict nightclub bouncer. It has extremely low permeability. The porins, which are the pores in the membrane, are very tight, making it hard for many drugs to get in the first place.
Okay, so a good defense.
And then it has built-in efflux pumps. These are proteins that sit in the membrane and actively spit antibiotics back out as soon as they manage to get inside.
So the drugs can't get in, and if they do, they get thrown right back out.
Correct. That's its baseline. But then it has acquired resistance, which it gets via plasmids. It can pick up genes from other bacteria in the hospital sink, for example.
And then there's the third type.
Yes. The most insidious type, the one that really burns clinicians, is adaptive resistance.
How is adaptive resistance different from just acquiring a new gene?
This is a resistance mechanism that is induced by the environment or by the treatment itself. The classic example involves the AMP-C beta-lactamase.
AMP-C. I've heard the story. You treat the patient, they seem to get better for a day or two, and then they crash.
Here is the why behind that story. The Pseudomonas chromosome has the AMP-C gene, which codes for a beta-lactamase enzyme, but it is usually repressed. The gene is turned off.
So it's there, but it's sleeping.
It's sleeping. However, if you expose the bacteria to certain beta-lactams, specifically those that are known to induce this gene, like ceftazidime or imipenem, that exposure can act as a trigger. It de-represses the gene.
So the antibiotic itself wakes the sleeping giant.
Exactly. The bacteria that survive the initial hit start mass-producing AMP-C beta-lactamase, which then hydrolyzes and destroys the antibiotic. You essentially select for the mutants that have turned on this defense.
This leads us to the golden rule of treating serious Pseudomonas infections, then. Because of this adaptive capability, you rarely fight it one-on-one.
That's right. For serious infections, pneumonia, bacteremia, we generally use combination therapy. You hit it from 2 different angles to prevent it from adapting.
What's a typical combination?
Typically, we use an anti-pseudomonal beta-lactam, like piperacillin tazobactam or cefepime, PLUS and aminoglycoside, like cubromycin or gentamicin.
2 different mechanisms of action. Overwhelm the defenses before they can adapt.
Precisely. That's the strategy.
All right, let's move into section 2, the clinical diseases. We've talked about the weapons, let's talk about the wounds. We've touched on the lungs, but let's solidify the pattern recognition here.
Pulmonary infections are the most common and probably the most serious manifestation. You have to categorize the patient into 2 main buckets: the cystic fibrosis patient and the hospitalized, mechanically ventilated patient.
Let's start with the CF bucket. What does the infection look like there?
In cystic fibrosis, this is a chronic, decades-long siege. The Pseudomonas usually enters early in childhood or adolescence. It colonizes the airways and sets up that alginate biofilm we discussed.
And once it does that?
Once it goes mucoid and establishes that biofilm, it is practically impossible to eradicate. Treatment becomes about suppression, not cure. It causes this chronic, smoldering inflammation that slowly, year over year, destroys the lung tissue, leading to bronchiectasis and eventual respiratory failure.
And the second bucket, the hospitalized patient.
This is ventilator-associated pneumonia, VAP. Here, the endotracheal tube is the highway for the bacteria.
How so?
It bypasses all the natural filtration of the nose and throat, and it delivers the bacteria, which may be colonizing the ventilator tubing, straight down to the lower lungs. And in a patient who is already critically ill.
It's devastating. It
causes a necrotizing bronchopneumonia. The tissue is literally dying.
What does that look like on an X-ray?
It's often bilateral, diffuse, with patchy infiltrates, and you can see areas of cavitation because of the necrosis from the elastase and other toxins. The mortality rate is staggering, historically up to 70% in untreated or poorly treated cases.
Moving from the lungs to the skin. We mentioned burn wounds earlier as a classic setup.
Burns are the perfect environment. You've lost the primary skin barrier, and the wound is moist and protein-rich. It's a feast for Pseudomonas.
And it doesn't just stay on the surface.
No, it colonizes, and then it invades the underlying tissue and blood vessels, thanks to elastase. This leads to vascular damage and systemic sepsis. The warning sign is a discoloration of the burn eschar. It often turns greenish or black,
Right.
and rapid tissue destruction underneath.
But there's also a skin infection that a perfectly healthy person can get, the hot tub rash.
Yes, Pseudomonas folliculitis. This is a classic board question vignette. A patient comes in with an itchy, red, bumpy rash papules and pustules, often in a bathing suit distribution.
Why the bathing suit distribution specifically?
Because the fabric of the swimsuit holds the contaminated water against the skin for a prolonged period, which forces the bacteria into the hair follicles.
And why hot tubs? Why not just swimming pools?
It's about temperature dynamics. Pseudomonas loves warm water. It can thrive at temperatures up to 43 degrees C. And just as importantly, hot water breaks down chlorine much faster than cool water.
So if the hot tub isn't meticulously maintained,
The chlorine levels drop and it becomes a warm Pseudomonas soup.
that's a lovely image. What's the treatment for that?
The good news is, unlike the burn wound infection, this is usually self-limiting in a healthy person. You don't typically need antibiotics. Just tell the patient to get out of the tub, wash their suit, and it should resolve in a few days.
Let's talk about the foot, the osteochondritis of the foot. This is a very specific one.
This is very specific pattern recognition. The vignette is almost always a child stepping on a nail. But not just any nail. A nail that goes through a rubber-soled sneaker.
Why does the sneaker matter so much?
This is the why that helps you remember it. The foam and rubber sole of a sneaker retains moisture and sweat. It's a perfect, warm, moist incubator for Pseudomonas.
I see.
So when the nail goes through the rubber, it picks up the bacteria from that moist interface and inoculates them deep into the bone or cartilage of the foot.
So, nail through sneaker points to Pseudomonas. What if it's a nail through a bare foot?
Then you're more likely thinking about Staph aureus or environmental Clostridium from the soil. But the sneaker is the key clue for Pseudomonas.
Now let's discuss the pathognomonic sign of Pseudomonas sepsis. The skin lesion that tells you this patient is in very big trouble. Ecthyma gangrenosum.
This is a critical visual diagnosis. Ecthyma gangrenosum. It doesn't look like a simple infection. It starts as a red patch, maybe a blister, but it rapidly evolves into a necrotic ulcer with a black eschar in the center, surrounded by a red halo.
A black center. That sounds ominous.
It represents vascular infarction. Remember the elastase we talked about? It's destroying the blood vessel walls in the skin, causing hemorrhage and cutting off the blood supply. The tissue dies and turns black.
And who gets this? Is this common?
No, it is not common. It occurs almost exclusively in severely immunocompromised patients. Specifically, those who are neutropenic, meaning they have a critically low count of neutrophils, their main bacteria-fighting white blood cells.
So what would you see on a biopsy of the lesion?
You would see the bacteria swarming the blood vessels, you'd see the vascular destruction, but you will notably see an absence of neutrophils.
Because the patient doesn't have any to send to the fight.
Exactly. It's a sign that the immune system has completely failed to contain the invasion. A very poor prognostic sign.
Briefly, let's touch on the other sites. Ear and eye.
For the ear, swimmer's ear, or otitis externa. Again, water exposure, usually mild. But in diabetics or the elderly, it can progress to malignant otitis externa.
Malignant usually implies cancer. What does it mean here?
In this context, it implies aggressive and invasive. It's not just a skin infection in the ear canal. It invades the cartilage and bone of the skull base, damaging cranial nerves. It can cause facial paralysis. It's a true medical and surgical emergency.
And the eye. What's the scenario there?
Contact lens trauma. If you scratch your cornea with a contact lens that has been sitting in contaminated solution, Pseudomonas can get in. It releases those enzymes we talked about, and it can literally eat through the cornea in 24 to 48 hours.
That fast.
That fast. It can lead to corneal perforation and blindness very quickly. You do not mess around with a suspected Pseudomonas corneal ulcer.
It's absolutely ruthless.
Okay.
That covers the leader, Pseudomonas aeruginosa, but this is a deep dive into the whole opportunistic alliance. Let's talk about the deceptive cousins, Burkholderia.
Burkholderia is fascinating because taxonomically, it used to be classified as Pseudomonas. Genetically, they're very close cousins. But clinically, they have carved out some very distinct and dangerous niches.
There are two main players here that Murray focuses on, the Burkholderia cepacia complex and Burkholderia pseudomeli. Let's start with cepacia.
Burkholderia cepacia is actually a complex of at least 17 related species, sometimes called genomovars. This organism is the nemesis of the cystic fibrosis patient.
Wait a minute, I thought Pseudomonas was the CF nemesis.
Pseudomonas is the most common enemy in CF, but Burkholderia cepacia is often the most feared enemy. It can cause something called cepacia syndrome.
That sounds like a disaster movie title.
It feels like one for the families involved. It's a rapid, fulminant deterioration with necrotizing pneumonia and bacteremia. A patient with CF who was previously stable can suddenly crash and die within weeks of acquiring this bug.
And there's a social tragedy associated with this bug, isn't there?
There is. This is a key point. Unlike Pseudomonas, which is mostly acquired from the environment, Burkholderia cepacia is highly transmissible from person to person.
So one CF patient can give it to another.
Exactly. Through coughing shared spaces. This led to the heartbreaking implementation of strict segregation policies in CF camps and clinics. Patients colonized with B. cepacia are often kept separate from other CF patients to prevent its spread.
And there's an impact on treatment options too, right?
A huge one. Perhaps most devastatingly, infection with this complex often disqualifies a patient from receiving a lung transplant
Why is that?
because the post-transplant outcomes are so poor. The bacteria often hides out in the sinuses or other tissues, and even with the new lungs, it comes roaring back once the patient is on immunosuppressants.
Now, for the exams in the clinic, how do we treat it? Because I recall it's different from Pseudomonas.
This is the key differentiator you need to remember. Pseudomonas is generally resistant to trimethoprim sulfamethoxazole, or TMP-SMX. Burkholderia cepacia, however, is often susceptible to it. TMP-SMX is a cornerstone of therapy.
So, cepacia equals CF, social distancing, and you treat with TMP-SMX. Now, what about its exotic cousin, Burkholderia pseudomale?
This causes a disease called melioidosis, also known as Whitmore's disease. Geography is everything here. The vignette will almost certainly involve a patient who has traveled to or lived in Southeast Asia, Thailand, Vietnam, Cambodia, or Northern Australia.
I've heard this called the Vietnam time bomb.
Yes, for a good reason. It could have a massive latent period. There were documented cases of American soldiers from the Vietnam War who developed symptoms decades after returning home.
And where does it live?
It lives in the soil, and water ice patties are the classic source. People get it through inhalation of contaminated dust or through cuts in their skin.
What does the disease actually look like?
It's called the Great Mimicker. It can present as an acute fulminant sepsis that kills you in days. Or it can present as a chronic indolent infection that looks exactly like tuberculosis with cavitary lesions in the upper lobes of the lungs.
And it has a darker reputation as well.
It does. It's classified as a Category B bioweapon agent. It's relatively easy to grow and can be disseminated via aerosol, and it's highly lethal if not treated.
And the treatment.
It requires a long course of antibiotics, often an acute phase treated with something strong like ceftazidime or meropenem, followed by a long eradication phase where, again, TMP-SMX is key. Moving on to Section 4, Stenotrophomonas maltophilia. That is a mouthful of a name. The name tells you the story if you break it down. Stenos means narrow, trokos means feeder, has a narrow range of food. But maltophilia means friend of malt. It strongly oxidizes maltose.
Friend of malt sounds like a craft brewer. But this bug is no friend in the hospital. What is its niche?
Stenotrophomonas is the ultimate scavenger. It's an organism of selection pressure. It typically appears in patients who are already on very broad-spectrum antibiotics, specifically carbapenems like meropenem or imipenem.
Why carbapenems in particular?
Because almost all other important gram-negative Rods E. coli, Klebsiella, Pseudomonas are susceptible to carbapenems. They are the nuclear option in our antibiotic arsenal. But Stenotrophomonas has innate resistance to carbapenems.
It just laughs at them. How does it do that?
It produces a metallo-beta-lactamase, specifically the L1 beta-lactamase, which is zinc-dependent. This enzyme is incredibly efficient at hydrolyzing and destroying carbapenems.
So when you treat a patient with meropenem,
You wipe out all the competition. The E. coli, the Pseudomonas, and Stenotrophomonas, which was probably just hanging out quietly, says, "Thank you for clearing the table for me," and it takes over and causes an infection.
that is a classic setup. A patient in the ICU on meropenem for a week for pneumonia suddenly spikes a new fever.
You have to think Stenotrophomonas. It causes bacteremia and pneumonia, and it's often associated with contaminated IV catheters or even disinfectant solutions.
So how do we treat the friend of malt? You can't use the big guns because it eats the big guns.
You have to go back to an older, different class of drug. Once again, TMP-SMX trimethoprim sulfamethoxazole is the drug of choice.
I'm seeing a pattern here. Burkholderia and Stenotrophomonas both seem to be susceptible to TMP-SMX. Pseudomonas usually hates it.
That is a very useful clinical simplification for memory. If it's a non-fermenter that isn't Pseudomonas, you should definitely be checking if TMP-SMX is the answer.
All right, Section 5. Acinetobacter.
Specifically, Acinetobacter baumannii. This organism has risen to infamy in the last 2 decades.
It acquired a nickname during the wars in the Middle East, Iraqabacter.
Yes, it became a massive problem for soldiers returning from Iraq and Afghanistan with traumatic blast wounds. It would contaminate the soil and debris blown into the deep, soft tissue wounds, and it was incredibly multi-drug resistant.
What is its superpower? We said Pseudomonas likes water. What about Acinetobacter?
Acinetobacter breaks that rule. While most gram-negatives need moisture and die quickly on dry surfaces, Acinetobacter can survive on dry surfaces for weeks.
Weeks on a dry surface.
Yes. Bed rails, doorknobs, blood pressure cuffs, keyboards, even human skin. It's incredibly resistant to desiccation. This is what makes infection control an absolute nightmare. You can't just dry things out to kill it.
And biologically, it can be confusing on a microscope slide.
It's a coccobacillus. It's plump and short, not a long, slender rod. On a gram stain, they often appear in pairs and can look very much like kochi. It can easily be mistaken for Neisseria.
So how do you tell them apart quickly?
The oxidase test again. It's so important. Neisseria is oxidase positive, Acinetobacter is oxidase negative.
A crucial distinction. And what about its resistance profile?
It's bad. We're talking about extremely high levels of multidrug resistance, MDR. We often have to resort to colistin.
Colistin. That's the antibiotic of last resort, right? I feel like I hear that a lot.
It is. It's an old drug, a polymyxin. It essentially acts like a detergent, disrupting the bacterial cell membrane. We stopped using it systemically decades ago because it is very nephrotoxic, it damages the kidneys, and also neurotoxic.
So why are we using it now?
Because we had to. We had to bring it back out of the archives because Acinetobacter strains emerged that were resistant to every other safer drug we had.
That is a sobering thought. We are digging into the archives of toxic antibiotics just to survive these things.
It's a desperate measure for a desperate situation. It highlights the threat of antimicrobial resistance.
Finally, let's touch on section 6, Moraxella.
Moraxella catarrhalis. This is our respiratory diplococcus in this group. It is a strict aerobe, and it's oxidase positive.
It looks a lot like Neisseria too, right? Gram-negative diplococci.
Yes, on a gram stain they can look identical, but the clinical picture tells you what it is. Moraxella targets 2 very specific patient groups.
Who are they?
First group, the elderly and smokers, particularly those with underlying lung disease. It's a major cause of bronchitis and acute exacerbations of COPD, chronic obstructive pulmonary disease.
And the second group?
Children. It's one of the top 3 bacterial causes of sinusitis and otitis media or middle ear infections in otherwise healthy kids.
So if a kid has an ear infection, it's usually strep pneumo, Haemophilus influenzae, or more Moraxella catarrhalis.
The big 3 of otitis media. That's exactly right. One important thing to know about Moraxella for treatment is that almost all strains now produce beta-lactamase.
So plain penicillin or amoxicillin won't work?
They won't. You need to use a beta-lactam that's stable to beta-lactamases or a combination agent like amoxicillin clavulanate, which we know is augmentin, or a cephalosporin.
Okay. We have toured the entire gallery of opportunists, from the great-smelling Pseudomonas to the malt-loving Stenotrophomonas, the dry-surviving Acinetobacter, and the respiratory Moraxella.
It's quite the lineup of troublemakers.
Now, before we let our listeners go, we need to test their retention. It is time for our rapid recall.
This is where we solidify the knowledge. I'm going to give you a clinical vignette or a specific question. I want you, the listener, to pause and think of the answer. Then I'll reveal the answer and the why.
Let's do it. Question 1.
A 24-year-old patient with cystic fibrosis presents with worsening respiratory status. A sputum culture reveals gram-negative rods that form extremely mucoid colonies on the agar plate. What specific virulence factor is responsible for this mucoid appearance? Pause. The answer is the alginate capsule.
The mucoid phenotype is caused by the massive overproduction of alginate, which is an exopolysaccharide. This forms the biofilm that is the hallmark of chronic Pseudomonas infection in CF lungs. It's not just slime, it's a shield against antibiotics and neutrophils.
Got it. Alginate equals mucoid equals the CF biofilm. Question 2.
A patient with severe burns develops a wound infection with classic blue-green pus. The responsible organism produces a potent toxin that inhibits protein synthesis in human cells. What is the specific molecular mechanism of action of this toxin? Pause. The answer is inactivation of elongation factor 2, EF2, by ADP ribosylation.
The toxin is exotoxin A, ETA. It shares this exact mechanism with diphtheria toxin. By inhibiting EF2, it completely stops peptide chain elongation at the ribosome. No protein synthesis means cell death, which causes the necrosis in the burn wound.
The mechanism is key. Diphtheria and Pseudomonas both inhibit EF2. Question 3.
An immunocompromised patient on chemotherapy develops necrotic skin lesions with a black center surrounded by a red halo. This is identified as eczema gangrenosum. A biopsy and histology of the lesion would most likely reveal a profound absence of which specific cell type? Pause. The answer is neutrophils.
Eczema gangrenosum classically occurs in profoundly neutropenic patients. While the bacteria do produce toxins that can kill white blood cells, the primary reason neutrophils are absent from the lesion is that the patient's bone marrow isn't producing them. This lack of defense allows the bacteria to invade blood vessels unchecked, causing the black infarcted lesions.
Scary stuff. Okay, question 4.
A hospitalized patient who has been on the broad-spectrum antibiotic meropenem for 10 days develops a new bacteremia. Blood cultures grow a gram-negative rod that is oxidase negative and is noted to strongly oxidize maltose. What is the most appropriate treatment of choice for this infection? Pause. The answer is trimethoprim sulfamethoxazole, TMP-SMX.
The organism is Stenotrophomonas multifila. The clues are the oxidation of maltose, multifilia, and the fact that the infection emerged while the patient was on a carbapenem. Stenotrophomonas is intrinsically resistant to carbapenems. TMP-SMX is the standard of care.
Friend of malt, enemy of carbapenems. Question 5.
A soldier returning from a deployment in the Middle East has a severe wound infection involving a plump gram-negative coccobacillus. The organism is oxidase negative, and lab testing shows it is resistant to almost all antibiotics tested. What is the likely pathogen? Pause. The answer is Acinetobacter baumannii.
The history, Middle East military conflict zone. The morphology, plum coccobacillus, the oxidase negative status, which rules out Pseudomonas and Neisseria, and the high level of multi-drug resistance are the classic profile. And remember, it's the one that survives on dry surfaces.
Got it. Question 6.
Pseudomonas aeruginosa produces several characteristic pigments. Which specific pigment functions as a cytophore to bind and acquire iron for the bacteria from the host? Pause. The answer is pyoverdine.
Pyoverdine is the yellow-green pigment that acts as the cytophore. Pyocyanin is the blue pigment that creates toxic oxygen radicals. A good memory aid. Viridae is green-yellow, think nature, growth, and nutrition. Cyan is blue, think cyanide, poison.
That's a fantastic way to remember it. Pyocyanin poisons, pyoverdine feeds.
Exactly.
All right, question 7.
What is the specific chemical compound responsible for the sweet, grape-like, or corn taco-like odor of Pseudomonas aeruginosa? Pause. The answer is aminocetophenone.
Well, it's often just described as grape-like. Knowing the specific compound helps solidify the unique biochemical distinctiveness of the organism. It's a good fact to have in your back pocket.
Last one, question 8.
Treatment of serious Pseudomonas aeruginosa infections like VAP or bacteremia usually requires combination therapy. A common regimen is an anti-pseudomonal beta-lactam plus an aminoglycoside. What is the primary reason for using combination therapy in this specific context? Pause.
The answer is to prevent the emergence of resistance, specifically adaptive resistance like the de-repression of AMPC beta-lactamase. While synergism can be a nice benefit, the primary driver for using two drugs upfront in empiric treatment is to ensure you have coverage and, more importantly, to prevent the selection of resistant mutants during the course of therapy. You hit it hard from two sides so it can't adapt.
A fantastic review that really solidifies the key points.
If you want to consolidate this even further, I highly recommend opening Murray's Medical Microbiology to Chapter 27. Specifically, look at Box 27.1, the clinical summaries. It's a great rapid refresh table. And take a look at the images of eczema gangrenosum. Once you see that picture, you will never forget it.
And as a final thought, remember the exceptions to the rules. That's where the points are on the exam and where the saves are in the clinic. Stenotrophomonas kills the carbapenem rule. Acinetobacter breaks the gram-negatives need water rule. And Burkholderia is the specific transmissible enemy of the cystic fibrosis community.
These organisms are the ultimate test of our medical defenses, both the patients and the hospitals. They find the cracks in the armor.
So, to all our listeners, keep your armor strong, wash your hands, and watch out for the green pus. Stay curious, stay distinct, and don't let the opportunists win. Thanks for listening to this deep dive.